Perfect.
Good morning. My name is Earle Holsapple. I'm the president of SciTech Development, and I'm here to talk to you about a new drug delivery combination. And with me is Brian Leyland-Jones, our Chief Medical Officer.
Good morning, all. I will make some comments at the end.
We appreciate the opportunity to talk to you about this novel new drug delivery combination that has enabled a very successful anti-cancer agent called fenretinide. Today, we're going to add a little surprise along with it. We believe that there's the opportunity for this new formulation to also have impact on COVID-19. But let's start with the first part. Obviously, we have to have the standard safe harbor statement. We're talking to you about an opportunity where we're raising $3.5 million to conduct a clinical trial for this reformulated drug in the treatment of T-cell lymphoma and subsequently small cell lung cancer or breast cancer. It's a unique opportunity because we have IND approval as of December. We have orphan drug designation. We're anticipating fast track designation down the road. Our intellectual property is issued throughout the Western world. This is not a standard study. This is a confirmatory study showing that the drug works in its new formulation. It's already been shown, the API has already been shown to be safe and efficacious in prior trials. It's a low-cost product, so the margins should be significant for those who bring this to market. Time to market is unusually short, so we ask you to pay attention to why that's true. And the market opportunity is vast because of the number of cancers that have shown potential in responding to this drug. So we're raising approximately $2.5 million to $3.5 million now to conduct phase 1A to confirm the safety of the product and arrive at the appropriate dose, and then the $12.5 million to complete the study through phase 1B. What have we created? We have created a proprietary drug delivery system that's a nanoparticle. Think of this as a small onion with the drug encapsulated between the layers of the onion. This camouflages this hydrophobic product where we have not been able to achieve bioavailability so that the product can circulate through the body and arrive at the cancer cells to achieve the therapeutic. We combined it with fenretinide, a well-known anti-cancer agent that's been hydrophobic. It's an interesting compound because it has both direct and immune effects on cancer cells, multiple mechanisms of action. It's been de-risked because it's been studied in dozens of clinical trials, the largest being one conducted in breast cancer prevention by Johnson & Johnson. So we have combined the delivery system with the active pharmaceutical ingredient to create a powerful new drug that will be disruptive in the marketplace, initially targeting T-cell lymphoma because the best prior results, efficacy results, are in that. This is versatile in the sense that we also have a topical indication to complement the systemic for use with diseases like cutaneous T-cell lymphoma or other CAR-T. We anticipate that this drug could actually be a mop-up in the sense of preventing relapses and metastasis. Why fenretinide? It was first developed by Johnson & Johnson decades ago as a dermatology product. It failed as a dermatology product, so it was switched over by the National Cancer Institute to be a therapeutic because internal studies at the NCI suggested there was a strong possibility that it would make a marvelous therapeutic product. The National Cancer Institute sponsored dozens of studies and clinical trials and many studies, but they never overcame the hydrophobic nature of the product. We did so. What has history shown about the efficacy of this product historically? Well, the diseases you're seeing there, small cell lung cancer, neuroblastoma, ovarian cancers, and the various T-cell cancer indications have been shown to respond in clinical trials to fenretinide if only the dose levels could be achieved. The picture of a woman is a Sézary syndrome, cutaneous T-cell lymphoma patient who was about to die, had been on every other therapeutic option available. And when she went on an emulsion delivery system of Fenretinide, she went into complete relapse. That particular study showed other similar results that make the case for Fenretinide having significant potential as a therapeutic. How does it work? Well, first of all, as this nanoparticle is miniature relative to the size of cancer cells, so that the nanoparticle circulates through the system, arrives at the cancer cell, either fuses or is absorbed, generating apoptosis. It's done through various mechanisms of action that are both direct and indirect. The fascinating new thing about this drug now is that there is significant data showing that Fenretinide is active in treatment of dengue, Zika, West Nile, Modak, HIV, and so on, a significant number of viruses. Considering that we're now facing this coronavirus problem, we have studied this further, and the literature and experts in virology suggest that we should be testing this with COVID-19. So we're in the process of doing so. We're going to apply to BARDA for funding to test this in humans because it has both direct and indirect impact. An in vitro study is not sufficient. We must test it in humans. It's an ideal product for this time because we can get to market quickly if it works, because we already have IND approval for the T-cell study. We can probably accelerate that for use in this disease. So it's a fascinating new opportunity. Competitively, the ST001 is unique because we are delivering far higher concentrations of drug per unit of delivery. Therefore, it's a practical product. The emulsion study required a 24-hour infusion. Ours will most likely require somewhere between 3 and 4, and we're delivering about 6 times the dose level so that we can get to the dose levels and stay at the dose levels shown from prior studies to achieve the therapeutic effect. There are other drugs as listed here in the marketplace to respond to cutaneous T-cell and T-cell lymphomas, but none of them have the potential that this drug has. So that there are significant unmet needs in that space. The target market initially is T-cell lymphoma and the subsets of mycosis fungoides and Sézary syndrome. As I said earlier, the reason we're targeting that market initially is because the strongest data exists for patients responding to Fenretinide. There's also strong data suggesting that it will respond well in the treatment of small cell lung cancer, and so we'll pursue that. So the combination of those two markets potentially could equal a $2 billion market. But this is really what we're after. These are a partial list of the cancers that have been shown to respond to Fenretinide, and therefore our job is to go back in and prove that the new formulation ST001 will generate a greater activity than seen before in these diseases. And if it does, even in a portion of these diseases, the market potential will be huge. Now add on the possibility of COVID, and you can see that an investment in this opportunity is certainly a very unique opportunity. From the point of view of market protection, we already have Orphan Drug Approval from the FDA for use of this with T-cell lymphoma, where we get 7 years of market exclusivity from the time we receive NDA approval from the FDA, and we get significant R&D tax rebates, 50% of the cost of developing the product from the federal government as well. The list below shows the orphan drug indications that came from the prior list that we would be applying for orphan approval with those as we go forward. We have financial projections that suggest that the entire phase 1AB study of both T-cell lymphoma and small cell lung cancer would be in the area of $16 million. But this is going to be tranched over time. and we might be going for other diseases other than small cell lung cancer or subsets of T-cell. So this is a preliminary budget, but we've worked up a model that's extremely detailed and available for your review at a future date. So what we're doing is we're raising right now about $2.5 to $3.5 million to commence phase 1A to reconfirm safety of the API in the new formulation. And we expect to also see some form of clinical responses in that phase 1A. Then we will move on to phase 1B in T-cell lymphoma, and we'll require about another $3.5 million to complete 1B. And then the balance of the funds required would be for a 1B study of small cell lung cancer. So we have two at-bats with two diverse different cancers, one solid tumor and one blood disorder. So it's a great investment opportunity for a relatively modest amount of money from the perspective of drug development.
Thank you, Earle. I'll take over from here. So as Earle has already stated, the IND was approved in December last year. In terms of scale manufacturing, 90% of that is done. We have 3 contract manufacturers lined up. In fact, we were in contact with 2 of them in the last 24 hours, and they depend upon size and scalability. It's an exciting time for the drug. For the phase 1A clinical trials, we now have 3 possibilities. One is in CTCL, as Earle described. The second one is we are submitting a large breast cancer application. And the third one, as Earle described, is COVID. So whichever one gets funded first, that will basically be the phase 1A. That will seamlessly pass into the phase 1B. And when we see efficacy in the phase 1B, FDA has asked us to return to them for us to describe that efficacy and define the size of a trial required in order to go to NDA. And we anticipate that that would be relatively small. If necessary, we will go on to further registration trials in other indications. So Rush will take the lead in CTCL. If you look on this slide, you've got Rush, Northwestern, going from basically top left to bottom right, Rush, Northwestern, University of Michigan, which are all proximate. And so we could probably complete the 1AB trial amongst those. If it's going to be breast cancer, then I would take the lead, and that could be done either at Columbia or Stanford, and COVID will depend upon the particular sites. Next. Management team. Earle managed the incubator at Carmonos. He's been CEO of several medium-sized companies, raised a lot of money. Ralph is a dear friend. I've worked with him for 30, 40 years, a brilliant pharmacologist, and now he's director of the lab program at the NCI at Frederick. He is indeed an oncology drug national leader. Myself, I was trained in the UK and at Cornell and Sloan Kettering. I ran the developmental chemotherapy program at NCI, headed the cancer centers at McGill and Emory, and my expertise is in drug development and biomarkers. Mike Burns and Lou are basically the key operations people. They're the nitty-gritty people. Christine and Michael basically will help Earle and myself with the fundraising. Andrew is the CFO. Elizabeth Krause covers general counsel. And in fact, Mike Burns and Ali Moyen are looking after the obvious topical application of this. Technical board, I will just point out 2 people. One is Tim Kuzel, who's taking the head of the CTCL program based at Rush. And the second one is Mike Sporn, who is probably well known to many of you. Published over 600 papers. He ran the chemo prevention lab at NCI. He's been a professor at Dartmouth for many years. He's a world expert in retinoids, TGF-beta, and everything anti-inflammatory. Potential exits. We've had discussions with J&J since they initially developed the product. They have interest in basically relicensing this new form. Remember, there are two aspects on this. One is you have ST001, which is ready to go into multiple tumor types and COVID-19, but this is also a platform technology that can be used for essentially any hydrophobic drug. So I'm just going to leave this up. It gives the references to Mike Spawn. You've got Michael Young on here as well. We've already covered this, and Earle and I would be delighted to answer any questions.